so I'm going to back up a little. So we talked about large scale bringing in or exporting of molecules, and so we call that exocytosis if it's exiting the cell, and then endocytosis if it's bringing it in. And then there's three different kinds. There's phagocytosis, which involves bringing in solids like food, and then we have pino, which is French for drink, so pinocytosis is bringing in liquids. And then the last one is receptor-mediated endocytosis, and that's a special condition that we'll talk about. So you guys remember phagocytosis? We talked about it. So it produces pseudopodia, these false feet. What's the cytoskeletal component that produces these false feet? I asked you guys this last time. Hey, microfilaments, good. And that's composed of actin, so then it produces these. These are what basically a vacuole is, a very large vesicle, but it's the same thing, right? It's like the difference between a truck and a bus. It's just different size. And then here's a bacteria being ingested by an amoeba, or this could be a white blood cell too. And so, what kind of electron microscope took that image? Transmitting electron microscope. That's right, because you can see the inside, right? So if we want to see the inside, the small structure of the cell, we have to use a transmitter. If you want to look at the outside, then you use scanning. Excellent. All right. So next one is pinocytosis. Same sort of deal, except in this case it's taking in liquids. Again, it's showing you here. What kind of electron micrograph is this? Transmitting Y. There you go, you can see inside the cell. Alright, so the last one that we didn't get to cover when we last met is receptor-mediated endocytosis. So this is a unique system where the cell has a receptor, and that receptor binds a certain molecule, and when it binds, it initiates a protein called clathrin. And clathrin it's basically kind of a U shape, so it'll start building up in this area. You know, if you have a bunch of U shapes, eventually they're going to form a circle, right? So this thing will pull in the receptor and the molecule and create a vesicle in that manner using this clathrin, which is shown here in these black lines. And so an example of this would be how a white blood cell would ingest low density lipoprotein, or LDL. So on the test I might ask you, give me an example of receptor-mediated endocytosis. What would that be? So this would be, macrophages ingesting LDL, or LDL. Right, it's right there, LDL, low density lipoprotein. Right there. Right here, on top of this. So viruses can exploit this process and enter the cell in the same mechanism. Yep, that's exactly right. Alright, so any questions about that? Okay, so that is the conclusion of chapter 7. Any questions about chapter 7? Alright, I guess we'll move right along to chapter 8 then. Okay, so chapter 8 has to do with metabolism, and guys use the word metabolism a lot, right? That person can eat whatever they want, so they have a fast metabolism, blah, blah, blah. But that's really not the correct terminology. So scientifically, metabolism is the totality of energy and molecule transformation. So we split that up because there's two different kinds. One is breaking down molecules or breaking down energy, and that is catabolism. And then if you're building up molecules or building up energy, we call that, right, anabolism, like anabolic steroids, because ana means what? Remember we talked about anatomy? Up. So anabolic means to build up molecules. Alright. So metabolism, again, is just the totality of an organism's chemical processes. There's always pathways that are involved in metabolism. Like, you can't just suddenly convert all the energy into sugar and sugar into all the energy in ATP, because you would burst into flames. So you have to do it in baby steps. And like we said, so I might ask you on a test, what metabolic process is involved in digesting the food that you put in your mouth? Is that anabolism or catabolism? Catabolism, right? What sort of metabolic process would be involved with plants taking carbon dioxide and building that into glucose? Anabolism, good. So, and if you're, making proteins, what is that an example of? Right, if you're breaking down proteins. So I think you guys get it. Alright, so when we talk about catabolic reactions, all of these things can be broken down. Carbohydrates, lipids, and proteins. In fact, all of these are on your nutritional label, and you guys know that this is a source of calories, so that you can digest all of this and release the energy from it. But in the same token, you can build all of these molecules as well. And you can build them from scratch. So, if you sit around and ate carbohydrates all day, what would that get turned into? Would it stay carbohydrates? What does it become? It becomes fats, right? Lipids. That's how we store our energy. So you convert the carbohydrates into lipids, and so on and so forth. Alright, so this is an example of metabolic pathway. This chart shows the metabolic pathways for you breaking down molecules and releasing energy. So it includes all the different kinds of sugar pathways, the fat pathways, different kinds of lipids, and different kinds of proteins. So this one that's highlighted here, it's a dark color, this is the pathway that you use to break down glucose and convert that into the energy of ATP. So each one of these dots represents a different worker, and what do we call the workers in our cells? Enzymes. So each one of these is an enzyme. Here's glucose. There's an enzyme. The first step of this pathway, this enzyme, is called a hexokinase. You guys probably don't know what a kinase is, but kinases are important. The kinase, well, the name tells you what it is, kind of. What is it ended? So what do you think it is? It's an enzyme. And its job is to add a phosphate to a molecule. So what do you think a hexokinase does? So it doesn't add 6, it adds it to a where did we use that word before? Hexose, right? So glucose is a hexose, right? 6 carbon sugar. So hexokinase adds a phosphate to 6 carbon sugars. So what happened here? What's the difference between this and this? Added a phosphate, right? That's exactly what a kinase does. And then this enzyme right here, so that's this dot right here is what we call an isomerase. What's an isomer? It's not the same shape, right? It's the same parts. Meros means part, iso means same. So an isomer is the same parts but different shape. So if you looked at this, this is still a 6-carbon sugar, and in fact, if you counted all the carbons, oxygens, and hydrogens, you would find out that this is still C6H12O6. So what has this enzyme done? It's rearranged that molecule so that it's an isomer of glucose. Does that make sense? This is called glucose 6-phosphate. What do you think that is? There you go. And you can count it, right? 1, 2, 3, 4, 5, 6. Glucose 6-phosphate. So why does it add a phosphate to it? What does adding a phosphate to a molecule do to it? What does adding any molecule to something else do to it? Change its shape, right? We talked about adding phosphates to the motor proteins in actin. What does that do? Changes its shape and allows your muscles to move. And then in the last chapter we talked about adding a phosphate to the sodium-potassium pump. What does that do? Changes its shape and allows you to pump things against the gradient, right? And we call that active transport. So again, enzymes can add phosphates too to other molecules, and that changes its shape, and that helps speed up, enzymes speed up reactions. They're like, we used this example before, they're like a worker in a factory. You could sit there and throw a tire or have a machine shoot a tire at a car. Eventually it might bounce onto it, but if you have a worker that actually physically puts that on there, it's going to go a lot faster, right? So that's what enzymes do. They direct the reactions so that they go faster. And then do you guys recognize this circle here? We did it in the lab in the biochemistry of cells. Krebs cycle. That's right. So this is the Krebs cycle here. Occurs in the mitochondria. So for the next test, I want you guys to memorize every one of these enzymes. I'm just showing you this so you can see how complicated it is just to take sugar and break it down into ATP. And if any one of these workers in the factory quit, right, any one of these enzymes got a mutation in it so it didn't work, what would be the end result? No, but you wouldn't get any energy, right? You wouldn't get any ATP. Diabetes has to do with not being able to get the glucose into your cells, right? So let's think about this since you brought it up. Let's say that you woke up this morning and you made yourself a bowl of cereal, right? What kind of energy source is mostly in cereal? Carbohydrates, right? So you stick it in your mouth. This is what you guys learned in 181, and what happens? So amylase starts to break down the starch, the carbs into, no? I mean, it's acting on amylase, but what's the output product? Maltose, right? It's a disaccharide. You guys learned that in the lab? So then you swallow it, goes into your stomach, and what does your stomach do? Does it break the whole molecule apart? What does it break? It does hydrolysis. So it breaks the glycosidic bond, so now you have individual glucose molecules. And then it passes into your small intestines. So probably about lunchtime it's going into your small intestines. And so you've gone all night. You haven't eaten anything, right? That's why they call it breakfast. It's breaking the fast. And so what do you think your blood level of glucose is at this point? And then inside your small intestines, high. So how does the glucose get from your small intestines into your bloodstream? concentration to low concentration, it's diffusion, right? Glucose needs a channel, so it's facilitated diffusion. So you get glucose in your bloodstream, it flows around, your pancreas detects that you have glucose in your bloodstream, so it produces insulin. Insulin binds to the cells and tells them to open a channel to allow the glucose to come in. So that's where you have your diabetes, right? If the channels aren't made, the glucose stays in your blood, and that's dangerous. You need to clear that out of your blood into the cells. So let's say that here's the insulin receptor. Here's insulin. It comes in, it binds, sends a signal to open a channel for glucose to come in, and then glucose comes into your cell. Right? And that is the first step from this glucose. That hexokinase adds a phosphate to it. And so now you have all of those dots involved in that reaction. And that's where it starts. Which is what's on the screen. So we're going to go through all of these reactions whenever we study the next chapter, chapter 9, which is respiration. So you eat food and you breathe air to simply put it in this pathway to produce what? ATP. That's your energy source. Alright, so we've talked enough about energy that you guys understand energy. So what kind of energy is heat energy? It's kinetic, right? Because remember, the definition of heat is the total kinetic energy in a system. What kind of energy is sunlight? Kinetic, right? Because it comes from the sun. Otherwise, if it didn't move, you'd never get it. What about the energy in a candy bar and the sugars in a candy bar? Potential, right? What about the energy in a rock at the top of a hill? What about the energy in a rock rolling down the hill? Kinetic. Good. So we can convert energy from potential into kinetic. In fact, you did it this morning, right? So you ate your cereal. What kind of energy is in the cereal? Potential. And then you walked into this room. So what kind of energy did that require? Kinetic. So you converted the potential energy in chemical bonds to the kinetic energy that allows your muscles to move so that you can come in here. So that was that a sigh? The energy conversion happens all the time, and that's required for living systems. So this is just an explanation of you eat the food, you slide down the ladder, so on and so forth. It's all interchanges of energy. There are scientists that study this. That field of study is thermodynamics. Alright, so in order to understand how reactions work, how your body's able to do this energy transformations, you have to understand a little bit about the chemistry involved in it and the reactions that are involved. So let's start off talking about two different kinds of reaction. One is a spontaneous reaction and one is a non-spontaneous reaction. So you guys know what spontaneous and non-spontaneous means, right? So what do you think a spontaneous reaction is? Just happens, right? And a non-spontaneous reaction just doesn't happen, I guess. So let's think about gas, gasoline. You guys are familiar with that, right? If I brought a gallon of gas in here and put it on the table, what would happen? No. So what kind of reaction is that then? Non-spontaneous. So, but you knew if I lit a match and dropped it in there, what would happen? It would become a spontaneous reaction. And the spontaneous reaction would continue until all the gasoline was burned up, right? Or someone put it out. But if you didn't interfere with it, that spontaneous reaction would go to completion. Does that make sense? Alright, so how did we get the non-spontaneous reaction to become spontaneous? Alright, so what did the fire do? What kind of energy? It's more specific. Heat energy, right? Or in other words, temperature energy. So kinetic temperature energy. Excellent. Alright. So thermodynamics explains how these reactions become spontaneous and non-spontaneous, and it can break it down into the exact numbers that are involved in this. So, paper burns at 451 degrees Fahrenheit. Gasoline ignites at a certain temperature, and so on and so forth. So there are certain laws or rules that have to be followed in this energy conversion. So we call these the rules of thermodynamics. There's actually 3, but I don't care that you know the other third one. Just you need to know the first 2 because that's relevant to biology. So the first one says that energy cannot be created or destroyed. It can only change forms. So what does that mean? Right. You can go from kinetic to potential, but you can't have more energy in the end than you started with in the beginning. Does that make sense? Right. So what does that mean? And Einstein took this a little further. You guys know Einstein's famous equation? E equals mc squared. What does that mean? Energy equals mass. That's the speed of light squared, right? But the important part of this is that energy can be converted into mass, right? And then the opposite of that is mass can be converted into energy. So this is the whole theory behind the atomic bomb, is that the mass of uranium or plutonium can be converted into pure energy. And this number is huge, right? So small amounts of mass equal massive amounts of energy, and vice versa. So, if you went all the way back and looked at the Big Bang Theory, what is that? What was in the beginning? Energy, right? If you believe the Big Bang Theory, there's a singular point of energy that exploded. And because of this law of thermodynamics, that means all the energy and all the mass in the entire universe that exists right now, because it can't be created or destroyed, was there in the beginning. And all of the matter and energy here will always be here. You can only change it into a different form. So I've got to tell you that this is the most tested law in all of science, even more tested than gravity, and it's never been disproven. Alright, so we can convert energy and light into chemical energy and chemical energy into heat and so on and so forth. Second law of thermodynamics says that every time you have an energy conversion, some of that order is lost. So an easier way to explain that would be, think about the pyramids, okay? What do they look like now? The Great Pyramids in Egypt. Do you think they look as nice as they did 2,000 years ago? Why not? It's because of the second law of thermodynamics, I hate to tell you. What about you guys? Are you getting better with age? You see like 60-year-old football players out there? Why not? Yeah. So because things get older, they break down, right? Is your car better than when you bought it? Can you think of any example of anything that gets better? Like we're talking physically, right? More ordered as time goes on. Still gonna get old, right? So over time, things tend to be disordered, right? And even when we do reaction, what? Right. Well, you have to put energy into that system, right? We're not talking about adding energy. We're talking about leaving it alone to its own devices, right? As things have energy conversions without any external input, they always go towards disorder. And that's another tested law of it's the second most tested law of all of science, and it's never been disproven. Yeah, everything gets old. Not more ordered. Yeah. So, yeah. Nobody in this room will ever disprove this tonight. I promise you. All right. No, it doesn't. It just thinks evolution means change in genes over time. Doesn't have to be better. Could be worse. All it means is you change, right? I mean, who's to judge if a chihuahua is better than a wolf? Doesn't mean better. All right. So I might ask you on the test, what's the first law of thermodynamics? Energy can't be created or destroyed. It can only change forms. What's the second law of thermodynamics? Every energy conversion leads to more disorder. And we can put a value on disorder. We can say that disorder is called.
Kind of like saying that you start dying daily.
Yeah, kind of. Things break down, right? Things age. So this disorder is called entropy. Right? So it has a name. And it also has a symbol, which is S. So the change, that, remember I wrote this on the lab, so that triangle delta is the Greek symbol and it means change. So this means change in disorder. Does that make sense? Alright. I think you guys get spontaneous reactions, right? Alright. So now let's talk about another thing. So there are two factors that determine if a reaction is spontaneous. I'm going to skip ahead some slides and I'm going to draw the Gibbs free energy equation. So this is Gibbs free energy, right? It's named after a guy named Dr. Energy. Scientist jokes. Tell you anymore. So Gibbs free energy, named after a guy named Gibbs, who developed this equation. This delta H represents another term which is on the board, which is enthalpy. This is a measure of the energy in a system which is available. that may be a little confusing, but this equation describes chemical reactions. Remember, whether or not a chemical reaction is going to occur on its own, spontaneously, or if it's non-spontaneous and we can get it to become spontaneous, like me throwing the match in the gasoline. Does that make sense? Alright, so let's just use what we were talking about before, so you guys know that you ate your cereal. So where's the energy in that? It's in the glucose molecule. So what is the formula? You guys eat sugars and you combine that with, what do you breathe? And that produces, what do you give off? And water. So this is the equation for respiration. We're going to go into this in great detail in the next chapter. This is what we're talking about right here. This mysterious arrow that somehow changes this into this. And the whole purpose of this is to do what? What are we making? What kind of energy? No. No. Yes, what kind? Well, I mean, where's it at? Yes. So your whole job, the reason you eat food and breathe air is to make ATP. I hate to tell you, that's it. Okay? So, that means that you're pulling energy out and storing it on ATP molecules. So what's the difference between these molecules and those molecules? Are there more of this? Are there more carbons? Are there more oxygens? Well, explain to me where's the energy? Why is this energy coming out of here? We can't create or destroy energy, right? It has to come from somewhere. So if we're making energy in the form of ATP, what's it coming out of? It's the energy in the bonds. So if we're talking about chemical reactions, then this part of the equation, the energy in a system that's available, because there's still bonds in here, right? So there's still energy in the system, but the energy we're releasing here is this, and it comes from the chemical bonds. Simple as that. Does that make sense? So, enthalpy is the energy in the chemical bonds. What do you think that is? What TV? That's right. And then what's delta S? Remember? Entropy, which is it's a measure of disorder. Alright, so the rule is, in order to get a reaction to become spontaneous, this value has to be negative. If it's positive, then what kind of reaction do you think it is? Non-spontaneous, that's exactly right. So, how do we get this to decrease? What can we do? Okay. Alright, so since you guys look a little confused, let's put some numbers in. Well, we just use this simple math. So let's say that this value is 1, and we'll say 1 is equal to I've just done is I've taken a non-spontaneous reaction and made it spontaneous by increasing disorder, right? Or I could increase temperature, which is what I did with the gas. But there's a problem here. Why doesn't this work in biological systems? What happens if I increase your temperature? It changes the shape of your enzymes, and then what does it do? They fail, right? They don't have the right shape, they can't function. So we can't heat frogs and people and trees up and expect them to have to increase their biochemical reactions, can we? Chemists can, because they do it outside of the body, right? So if you take chemistry, you want to get a reaction to work, all you got to do is heat it up. It's so super simple. But in biological systems, that's hard to do. Can I increase disorder in a living system to make it work better? Why not? Oh, come on. We made a list of the requirements for life in the very first class. What was one of those? Order, right? So disordering something goes against the requirements for life. So you can't do either one of these. As a biologist, as a chemist, sure, no problem. But as a biologist, no, you can't do that. So what's the only thing that we can change to get a non-spontaneous reaction to become spontaneous? So I can lower this. So let's go back to this. Say this is 1. If I make this 0, right, I'm reducing it, then what do I have? I've made a non-spontaneous reaction spontaneous. And that's the only thing that I can mess with in a biological system. So what do I have to do to decrease enthalpy? What's enthalpy? So what do we do? So we have to not really reduce the number of bonds, right? We have to. This is the energy in the bonds. So what do we have to do? Weaken the bonds. How do you do that? No. Here's a crayon. That's like a chemical bond, right? Weaken it. There you go. So what did she just do? She bent it to break it. She didn't pull it apart like this, did she? So what can we do to bend the bond? No? That makes it stronger. What did you just do? What's that? What's this? What's that? Are you changing your shape? How do you change the shape? No, you can't. Temperature will kill you. No, that will kill you too. When we add a molecule, a phosphate to another molecule, what does it do? Changes its shape. So enzymes can take a phosphate off of ATP, stick it on a molecule, cause it to bend, and weaken the bonds. They don't do it, right? It's the phosphate that's doing it. But they're the workers. They facilitate it. They're like, okay, I'm going to take the phosphate off of here and stick it on the molecule. It's going to bend it, and it's going to cause this to go down, and now I have a spontaneous reaction. So does that make sense? It's pretty cool. Hey, the screens are still on too. That's pretty cool. Okay, thank you for breaking my crayon. So I'm just going to skip ahead because I kind of just basically went over all these slides. So if we have delta G is negative, that's a spontaneous reaction. Remember, so I'll give an example of something that's done in the laboratory and in your body. So you guys know that in your body you have to copy your DNA, right? In order to copy your DNA, what has to be done? Remember, it's double-stranded. It's held together by what? What holds DNA together? Hydrogen bonds, right? So how do you get the hydrogen bonds to come apart inside your body? That would chop up your DNA. How do you get this to be spontaneous, right? Well, you change its shape, right? So you just twist it the other way. So there's an enzyme called helicase, and that's what it does. So it will break the bonds. Can you heat up inside your cell to make your DNA come apart? That would mess up everything, right? Can you make it disordered? So hydrolysis, no. But if you take DNA out of a cell, right, like Jerry Springer or Maury Povich does to the baby daddy, and you want to copy that DNA in a test tube, how do you think you do it? What's the easiest way to do it? To get the DNA to come apart? Yeah, you increase the temperature. So we increase the temperature almost to boiling because it's not inside a cell anymore, right? We don't have to worry about it. So chemists can use this, and biologists do too when it's not in a living system. But when it's inside a system, we're forced to use this. But anytime it's outside of a system, this is much easier. Alright. So, any of you guys had chemistry before? So what does a chemist call a reaction that releases energy? Or what'd you say? Exothermic, that's exactly right. So a chemist would call a reaction exothermic. What does thermi mean? Temperature, right? Is the chemist right? Why? Yeah, because it doesn't have to be temperature, right? Chemists are biased because they don't have to work with living systems. So this is usually what they use. But in reality, it's energy that causes the reaction. So the correct term, if I were to discuss this with a chemist, I would say it would be exergonic, because that describes the release of energy, not the release of temperature. Does that make sense? But it's basically the same thing. It's just semantics, although chemists are wrong. So this describes this reaction, right? I mean, you guys know that the energy in the reactants has to be bigger than the energy of the products. Otherwise, we wouldn't get any energy out of it, right? So this graph is showing you that an exergonic reaction, the reactants have more energy than the products. And remember, the law of thermodynamics says we can't create or destroy energy. So if this has more energy than that, where does the energy go? It has to go somewhere else, right? It has to go out of the reaction. And in this case, it goes out and into ATP, right? And so the difference between these two The energy available in the reaction is the free energy. So what does this line represent? Delta G. Does that make sense? You know, and is this spontaneous or non-spontaneous? It's spontaneous. Delta G is negative, right? Rocks, like this graph, rocks roll downhill. Do they need energy in them? A rock rolling downhill. So let's just say the rock's rolling. Do you need to put energy in it to get it to roll, or to keep it to rolling? So the graph looks like it's a hill, right? You want to go from here to here, then you're going to release energy. You don't have to use that example if you don't want. You can just memorize it. But I think it helps. Alright, so if we put numbers into the equation for this, you would see that 1 mole of glucose, remember a mole, that has 600 so it's negative, right? Because it's releasing energy, so it's negative 686 kilocalories in 1 mole. Remember, we measure our calories as kilocalories. So if you ate a mole of sugar, how many calories would that be? 686 calories. And so remember, if that's how much you release, and plants do the opposite, how much energy do you think plants have to put in order to make a mole of glucose? According to the law of thermodynamics, it has to be the same, right? So it's positive 686. This energy can't be created or destroyed. So this is the opposite reaction. This is what plants do, right? So they take molecules that have less energy in it. So now, if the arrow's that way, what are these called? These are the reactants, these are the products. So what has more energy, the reactants or the products? Products. So this drawing showing that the reactants have less potential energy than the products. So remember, energy can't be created or destroyed. So how do we get this to be that? We have to put energy into the system, right? Where does the energy come from? This is a photosynthesis, so where does the energy come from? Sunlight, right? And that's what drives the reaction. So here's the difference. What does this arrow represent? No, the difference is the delta G. Is this delta G positive or negative? It's positive, right? So is it spontaneous or non-spontaneous? Non-spontaneous. So enzymes are required to make it spontaneous. And we call that reaction endergonic because it takes in not necessarily heat, right? Because that would be exothermic. I'm sorry, endothermic. But this is energy, so it's endergonic. Requires energy. And so you guys guessed it, 686 calories of energy have to be input in order to produce 1 mole of sugar. Because the law of thermodynamics says that we can't create or destroy energy, we can only change forms. Alright. Okay, the screen went off. I see it, the thing is blinking. Okay. So, any questions about that so far? Okay. So, now we talked a little bit about the chicken egg, right? We can affect the order of a system by inputting energy. And in fact, this delta G can never be zero, because if it was ever zero, then there would be no exchange of energy. A good example that your book has is if we have a closed system of these water tanks. I'll give that one a second to come on. So we have this closed system of these water tanks, and we have more water in this tank than this one. Then the water's going to flow this way, right? Spin this turbine, create energy. You guys know that's how a dam works. So but eventually, if this is closed, the water levels are going to be the same, and that's the same as delta G being zero. So can you produce energy when delta G is zero? So then what happens to life then? No energy. Remember we said the very first day, one of the requirements of life is you have to be able to utilize energy, right? You need an energy source. So we would be dead. Why aren't we dead? Because we're not a closed system, right? If I put you in a box, you would die eventually, right? But you're not in a box. So where do you get your external input of energy? Well, yeah, where does it get its external input of energy? The sun. Yeah. So it's like if you opened up this container and allowed water to flow in it, as long as that water's flowing, it keeps this at a disequilibrium, and so delta G is never zero, and so life can go on. What happens if the sun burns out and we lose our tap? Yeah, life stops. So this inequality of delta G being positive or negative and fluctuating allows life to exist. And that's why we're allowed to exist. Okay, so that brings us to the magical molecule ATP. So you guys know that if we take a phosphate and add it to a motor protein, it can move our muscles, it can move organelles around. If we take a phosphate and we put it on integral membrane protein like the sodium potassium pump, it can change its shape so we can pump it against the gradient. You now know that if we add a phosphate to another molecule, we can make a reaction occur, right? So where do all these phosphates come from that are being added left and right in your body right now? They come from ATP, right? Adenosine triphosphate. So that ATP is like a spring, like a coiled spring, so it has energy in it. Kind of like a BB gun. So if I put a ball on the end of a spring and I pushed it down, that has potential energy, right? What if I let go of the spring? Then it will shoot the ball off, but if I put another one on there, will it work? No. So how do I get it to work again? I gotta compress the spring again, right? So ATP is kind of like a compressed spring. It has energy in it. And that energy comes from these three phosphates next to each other. So you guys know that that's adenosine, that's an RNA nucleotide, same one that's in A in DNA, but the sugar's RNA. And then that stands for tri, and that means phosphate. So when this thing adds a phosphate or shoots off a phosphate, then it's like an uncoiled spring. And so if we lose a phosphate, what does it become? Diphosphate. So this is like a uncompressed spring. So if I want to recompress this, what do I have to do? Add a phosphate to it, and then it gets compressed again. So that's what your body does. It takes ADP and reloads it with phosphate so that it can use it to do work. You guys know that it can use phosphates to do all kinds of stuff. So mechanical work, we talked about that, contracting muscle cells, transport, sodium, potassium pump, chemical, right? Doing reactions, like changing the delta G. So all of that is done by ATP. So here, this is ATP. There's the base, right? You guys know, what kind of base is this? Purine or pyrimidine? I'm not going to tell you, I'll make you look it up. So here's a sugar ribose, right? Here's the phosphates. Why is this unstable? Why are 3 phosphates like a compressed spring, ready to throw that phosphate off? They're a bunch of negatives next to each other. So if you have a bunch of negative magnets next to each other, what do they want to do? They want to repel from one another. And it's the same thing that's going on here. Alright, so just like all the other reactions, and I asked you guys to do this when you did the chemistry lab, but you probably forgot because it was so early in the semester. When I asked you guys to take a adenosine triphosphate and then split it into diphosphate, remember that? So if you want to take ATP and make it into ADP, what do you have to do? Add water. What kind of reaction is that? Hydrolysis. And you want to reverse that, what do you have to do? Take out water, and that's dehydration. So that's the reaction to make that. The reaction has 7.3 kilocalories per mole in it. And so we can use that to figure out how efficient your body is in converting the energy of glucose into the energy of ATP. And we'll do that when we get to chapter 9. So you guys use a lot of ATP. Do you know how much you use? You use about 180 pounds of ATP per day. So you think you eat all that ATP? You think you manufacture all that ATP? So how do you use so much ATP? You're recycling, yeah. So you're just recycling this ATP to ADP and then back to ATP. So all you have to do is just reload the phosphate. Alright, so we just talked about this, how it adds a phosphate and changes its shape to reduce delta H, which makes delta G negative, which makes the reaction spontaneous. But here's an example of how this really works when we just put in some numbers and we do some mathematics on it. So remember the equation, I didn't give you guys numbers, but here I will. So this is a reaction your body requires. It takes glutamic acid and combines it with ammonia, and that makes glutamine, which is an amino acid. Okay? So you have to synthesize this amino acid, and the delta G for this reaction to add glutamic acid to ammonium is plus 3.4 kilocalories per mole. Would that reaction ever happen? Delta G is plus 3.4. Would gasoline ever just ignite by itself? So this would never happen. So how do we get it to happen? Add a phosphate, right? Remember, that changes its shape, bends the molecule. So how much does it bend? Well, the phosphate has the energy, right? And the energy is 7.6 or 7.3 kilocalories per mole. So if we have plus 3.4 and we add negative 7.3, what does that make the delta G? So what's the reaction now? Spontaneous. So all you've done is add a phosphate, but you've done it in such a way that the delta G is now a negative value. Yes. Thank you for letting me know. Okay, so you guys know that ATP is regenerated, it's recycled. ATP goes to ADP, and then we add a phosphate back to that, and we make it recharged. So we talked about you go through 180 pounds, but in every single one of your cells, in a working muscle cell, 10 million ATP are consumed and regenerated per second per cell. 10 million per second. It's a lot. It's a lot of energy. All right. Any questions about anything we covered so far? Right. Just adding a phosphate. Okay, so you guys all did the enzyme lab, and you guys understand that enzymes are proteins, right? So they're made out of amino acids, and they can be affected by temperature and pH. Enzymes are what we call catalysts. So a catalyst is something that speeds up a reaction without being changed or consumed in the reaction itself. Just like a worker in a factory, right? Just because you put the processor in your iPhone doesn't mean that you become part of the iPhone, or that you can't do another one again after you put one in. So enzymes are like workers. They can do the same job over and over and over again. Eventually they get tired and they wear out. And why do they wear out? Second law of thermodynamics. Like, everything gets old and broken down. Alright, so enzymes do not change delta G, right? They're just like a worker doesn't really become part of your phone. So if they're building it. So enzymes only dictate the phosphate being added to the molecule. Does that make sense? It's the phosphate that's changing the delta G, not the enzyme. So if I ask you on the test, the delta G of a reaction is plus 20 kilocalories per mole, and I add an enzyme to it, what's the new delta G? It's the same. It's plus 20 kilocalories per mole, because enzymes do not change delta G, right? That's important. It's the phosphate that changes delta G. It's the changing of the shape of the molecule that changes the delta G. Enzymes just help speed up reactions. And you guys know enzymes end in ASE, hopefully. Alright, so I kind of lied to you earlier about the rock, and that you kind of caught on to that, right? So I was like, oh, you push a rock downhill, and do you have to put any energy into that? Well, you guys know, the rock just doesn't suddenly start rolling downhill. You have to give it a little nudge, and chemical reactions are the same way. So we call that little nudge activation energy. And so it's a little hump that I didn't show you before. So the graphs I showed you before were from here, and they went straight down. So now we're showing you that there is a little hump. You have to get it to start going, and so that's called energy of activation, or activation energy, and it's usually symbolized as E sub A. And this is what enzymes do, is they lower the amount of energy that you have to put into the system to get it to go. They give you a little help in shoving the rock down the hill. And the point so in a chemical reaction, and the artist is showing you this, so we have A molecule bound to B. It could be like this, right? And then we have C to D, so these are oxygens bound together. And if you want to take this and create this, what do you have to do? Well, let's just say I gave you this molecule in a model form, and I said make that. What would you have to do? do you have to do? You'd have to take it apart, wouldn't you? You'd have to break the bonds and then rearrange them into this. So that's what happens here in the transition state is you have to break these bonds in order to reform them. So that's what we call transition state. Alright, so enzymes speed up the reaction by lowering the activation energy, which you can see here. So it just makes it easier to get over that little hump, that transition state. Remember, enzymes don't change delta G. They only change what? Activation energy. Any questions? Okay, so just a reminder, next week, on Tuesday, there's no lecture, right? You guys have a study day. But there will be a lecture on Thursday. But for my class, just my lab, you guys don't have to come in for lab. You're going to do the mitosis lab at home. And you guys that have Tuesday lab, you're going to come in and do the exact same thing that they're going to do at home here. I think, right? So all of that's in the syllabus. I'm just reminding you. So I'll see you guys again on Thursday. The second exam is on chapters 6, 7, 8, and 9. All right, have a good weekend.
I have a question for you.
Yes.